Yes, good afternoon. I welcome you to my third lecture, which is related to the blueprint of life. The blueprint of life is the nucleic acids. Nucleic acids like DNA and RNA. How DNA and RNA look like, their structural features, as well as we shall discuss how they are a blueprint of life. Uh, so this is very important and interesting topic. If you and it is the a key topic in molecular biology. If you understand the structure and functions of the blueprint of life, that means the DNA, RNA, how they are functions in the cell, then it would be a good, base for understanding the biotechnology. So all living things are made of, water, because life originated in water, and water is an inorganic compound. And other inorganic compounds, mostly salt. So water and salts in the sea, it is believed that life originated, single-celled life originated there. And then organic compounds contain carbon and hydrogen, carbohydrates, lipids, proteins, nucleic acids. They are the organic compounds. So for any living organism, you can find water, you can find inorganic compound, you can find organic compounds. And organic compounds must be present in the living organism. Organic compounds means compounds biosynthesized in the living cell. So any compound, any plant or any living organism, when you analyze, you can find at least carbon, hydrogen, oxygen, and three elements in very high quantity. For example, if I ask you to analyze a plant leaf, how much you will get carbon, how much you will get oxygen, how much you will get hydrogen in weight or the amount in mass. So it is very important question as a biologist. I'm not sure you know it or not. Naturally, in living organisms, especially in plant, 45% you will find the carbon, nearly 45% oxygen, and around 5% hydrogen. So total 95%. So rest all other essential elements you can find only 5%. So carbon, hydrogen, oxygen, these three elements are synthesized in the living organism mainly through a process called photosynthesis. So photosynthesis synthesize the carbohydrate, carbon containing organic compounds, and they are used as the raw materials for generating energy as well as synthesizing the other compound, including nucleic acids, protein, and lipids. So you must remember and understand that any organic life, these three elements are largely present. When we look at a living thing, what we can see? We see mostly protein. For example, your hairs, your skin, your muscle, everything is protein. So how does an organism produce its particular protein? This is a big question. In the cell, mainly proteins are the main workers in the living organism. So protein synthesis is very important. As in people protein versus tree protein, they have the huge differences. So uh, uh, the answer of how does a protein is synthesized, the answer is DNA. That means DNA is involved in protein synthesis. And proteins are the workers of any living activity of a cell. So DNA, the species particular DNA sequences produce the species particular proteins. So DNA sequence for specific any species, even within the species, different individuals are buried. So we have now 7.3 billion people in the earth. The DNA sequence of all the living humans, they are different, slightly different. And genes code for proteins. So DNA contains some small unit which we call the gene. When gene is expressed, then protein is synthesized. We shall discuss this terminologies more in depth. Genes are long strand of DNA on chromosome. That means genes, DNA is a very long strand, double helical structure, and gene is small fragment. That means one DNA contain lots of genes, and genes carries the secret secret code that carries the information, and expression of the gene is associated with synthesis of the protein. How genes and DNA are involved in synthesis of the protein, we shall discuss in more detail. And now, interesting question is DNA. You know, DNA is basically nucleic acid. DNA is a chemical substance. Although it is the secret, it contains all secret information of a living organism. Nucleic acids are macromolecule. That means molecular weight is big. It is a bigger size molecule. Polymer occurs in every living cell, where they not only are responsible the storage and transmission of genetic information. This is very important. They store, for example, I am like my father or parents because I got DNA from them. So DNA store the information characteristics and then transmit through hereditary process and translate this information for the synthesis of protein. And now I am talking and lots of gene is expressed so that I can talk and deliver the lecture, and they are located in the DNA. Nucleic acids makes up the blueprint of life. This is very, very powerful statement. So nucleic acids are highly organized polymer. They are polymer. Polymer of what? What? Nucleotide. Small unit is nucleotide. For example, protein is a polymer. If I ask you protein, what is protein? You can say polymer. Polymer of what? Polymer of amino acids. For synthesis of protein, 20 essential amino acids are present, you know. So nucleotides are the unit of the nucleic acids. What is DNA? I am sure you have studied a lot in your biochemistry course, but for giving you a review, DNA is a genetic code, and DNA instructs for heredity, composed of genes, director of protein synthesis. So DNA is executive director, like our Sheikh Hasina. Sheikh Hasina does not need to do the all the job in the country, but she is the director, chief executive. DNA is like that. DNA remains in its location, but directs for protein synthesis, and proteins are the real players to do the job. DNA is also a type of nucleic acid, a type of organic compound, obviously organic compound because it is biosynthesized in the living cell, a polymer, a compound made of repeating subunits of nucleotide, and DNA stands for, what DNA stands for? Deoxyribonucleic acid. Deoxyribonucleic acids consist of a ribose sugar. Later I shall show you how ribose sugar look like. With a missing oxygen, that is deoxy. Deoxy means missing one oxygen in the ribose sugar, and it's found in the nucleus of eukaryotic organisms. Eukaryotic organisms, multicellular organisms or unicellular organisms where nucleus is enveloped. But bacteria, archaea, they are not eukaryotic organisms. But blue-green algae, human, animal, they are eukaryotic organisms. Where DNA locates, the simple answer is they locate on the chromosome. Where chromosome locates? Chromosome locates in the nucleus. Now, you may ask, does DNA locate only in the nucleus? The answer is no, because DNA can be found in the mitochondria in human. So most of the eukaryotic organism in nucleus and mitochondria DNAs are located. But in case of plant, DNA locates in another place called plastid. So plastid DNA, mitochondrial DNA, and nuclear DNA. 3 types of DNA present in the plant. So plant has 3 locations of DNA. How does DNA code for protein synthesis? This is a simple, I shall discuss later on. DNA has, 8 in different order. These bases are, present in the phosphodiester, framework, and DNA, 3 letters, each 3 letters can code for an amino acid. And when a gene is expressed, then messenger RNA is synthesized and they move to the ribosome, and this is the ribosome, a cell organelle where protein is synthesized. I shall discuss more detail on this topic because central dogma is, your one of the important course content. DNA and protein synthesis, then what happened? DNA sequence codes how letters are put together, produce messenger RNA sequence code. So, and then, messenger RNA leaves the nucleus. Then messenger RNA, the messenger, truly messenger, moves from the nucleus, that is office of the DNA, and go out to outside the nuclear membrane and moves to the ribosome, and present in the cytoplasm of the cell. And in the ribosome, the message it carries, they recruit, tRNA, recruit the amino acids, and amino acids one by one, depending on the, instruction message given by the DNA, they integrate and make a polymer of protein, and protein is synthesized. So transfer RNA brings amino acids depending on the message carries by the mRNA to the ribosome to match the sequence codes. So this is the simplest summary of today's lecture. We shall discuss more in detail. DNA, let me finally summarize. DNA is a nucleic acid. DNA stands for deoxyribonucleic acid, polymer of covalently linked deoxyribonucleotide units, and lack of oxygen at 2' of its ribose sugar. So this is the basic properties of DNA, and DNA has some functions. Already you know that chemical basis of heredity, that is transfer of properties from parents to the offspring, and may be regarded as the molecular repositories for genetic information. And DNA is not only, basis of heredity. The activities of any organisms, living organism, the behavior, traits, all are associated with the secret code and information present in the DNA. For example, white people, they have the blue eyes, and we have the black eyes, we have black color, they have the blonde, that is golden like hairs, due to the differences in the DNA sequence. So DNA is exclusively responsible for maintaining the identity of different organisms over million years. So human remains as human after million years due to the DNA is carrying the the properties. RNA is the cousin of DNA. RNA is the cousin of the DNA. RNA stands for ribonucleic acids. RNA can be different types. So RNA is the genetic material carrying hereditary information in many viruses. But for example, COVID-19 virus, it has no DNA. Many viruses in the earth, they have only RNA. And RNA also can carry the genetic material. And when DNA express, mRNA, carries the information. So RNA can be messenger RNA, rRNA, tRNA. So RNA as ribonucleoprotein participates in the post-transcriptional modification of other RNAs. So RNA can be different types, and different RNA can do different job. And in biochemistry, you studied in detail, and you can get here. So I am not going detail more. So nucleotide, you know that nucleotide is the phosphoric acid ester of nucleic acid. What is nucleoside? Nucleoside means without phosphoric acid. That means a nitrogen base, a pentose, one or more phosphate. These three unit together called nucleotide. But if it is one nitrogen base and one pentose together called nucleoside. So this is very simple. And nitrogen bases, in the DNA and RNA, purine and pyrimidine, two types of nitrogen bases are present. For example, purines, mainly adenine and guanine, and in case of pyrimidine, cytosine, thymine, and uracil. But in case of DNA structure, we can find only adenine, guanine, cytosine, and thymine. We never find uracil. Uracil is not present in the structure of DNA. But in case of RNA, uracil is present in instead of thymine. So RNA contains uracil. This is the chemical difference between DNA and RNA. If you are asked what is the chemical differences, structural differences between DNA and RNA, you can say DNA composed of adenine, guanine, cytosine, and thymine. And RNA consists of adenine, guanine, cytosine, and uracil, no thymine. And ATGC in the DNA, and AGCU in case of RNA. So sugar, I told you in the both nucleic acid, RNA and DNA. In case of RNA, beta D ribose is used. But in case of DNA, beta D2 deoxyribose is present. Here you see OH, here is only H. So this is the difference between the sugar present in RNA and DNA. And now this is final review. You can see DNA double helical structure present in the chromosome, and chromosome locates in the nucleic acids in the cell. And we have, do you know how many chromosomes are present in human? 23 pairs, and only one pair is the sex chromosome, but 22 pairs are non-sex chromosome. So chromosome and DNA replication is very important. Nucleus of one human cell contains approximately 1 meter DNA. You can imagine when you make it is a linear. But how they are present in the cell? They are DNA tightly wrapped around the protein, and it is called nucleosome. You can see histone protein, DNA tightly wrapped. DNA is not present a linear. Here you see DNA structure is tightly wrapped. Inside is the histone protein 3D structure. So DNA how DNA locates and present in the nucleus? It is wrapped around the histone protein, and it is called nucleosome. So DNA replication is necessary, because when one cell is divided to two, in that case, exactly same DNA should move to the other cell. In that case, DNA unzip, double helical structure unzip, and new nucleotides are synthesized and make new DNA. So when cell division taken place, before the, our new baby is born, carries the DNA from the parents. In that case, DNA replication is essential. So DNA replication is an important property for the DNA. So DNA can make its copy. And in the laboratory, by using PCR machine, we can make DNA copy. So must occur before the cell divides. Each new cell needs a copy of the information in order to grow equally. So how this is the DNA structure, parent DNA structure. So it separate into two single strand, and then new strand synthesize in the cell, new strand, and become one DNA to two. So this can be first divided, and then a new cell is developed. So RNA very similar to DNA. Already I told you, exception is ribose is 5-carbon sugar, no deoxy, and uracil replaces thymine, single-stranded. Another characteristic is that RNA is single-stranded. So you may be asked a question, compare and contrast DNA and RNA. So in that case, there are some similarities, there are some dissimilarities between RNA and DNA. One of the important dissimilarity is that DNA is double helical structure, as the picture looks, but RNA is single-stranded, not double helical structure. And who discovered the DNA? James Watson, Francis Crick, they published a paper, double helical structure of DNA in 1953. And Rosalind Franklin, one lady, and Maurice Wilkinson's group, he worked, and by using X-ray, they found that if DNA is double helical, then it must be X-shaped. So Rosalind Franklin shared the information with with her friend Watson, and finally Watson and Crick established the structure. So DNA is a double helical structure. For this big discovery, Watson, Crick, and Wilkins got the Nobel Prize in medicine. But Rosalind Franklin did not get, because unfortunately she died of cancer, breast cancer, before the Nobel Prize declared. But she is very famous because she first made the X-ray crystallography to confirm the real structure of DNA as double helical structure. And this is the very small, one-page paper published by Watson and Crick and Wilkins, molecular structure of DNA nucleic acids, on 25th April of 1953 in Nature, Weekly Nature Journal. And that was the, big discovery in biology, and the discoverer got the Nobel Prize. And biotechnology, or our better understanding in the biology, emerged or started from the real structure elucidation of the DNA. So first, Watson and Crick, they made this DNA double helical structure by using wires as well as the tin plate, and made it. And this is, you see, very nicely twisted structure. Now, the diameter of DNA structure is about 2 nanometer, and one twist to another twist around 3.4 nanometer size. So here hydrogen bonds holding this structure, different bases, for example GC, AT, and this double helical structure looks like this. So and it is a balanced structure, and A always bind with T, and G always bind with C, and never there is never any differences, and it is a rule. And why it is, I shall discuss later on. Here you can see hydrogen bond, how they connect guanine to cytosine and adenine to thymine. This is the chemical structure of DNA, and they are anti-parallel. You see 5 prime to 3 prime, here 3 prime to 5 prime, and this makes the DNA structure stable. And now let me briefly summarize the DNA and its building block. You see three units are present. One is base, another is sugar, phosphate, and another is nucleotide. So here you can easily understand. I gave the very colorful. First single strand is synthesized, and then recruit one by one the nucleotide monomer and make the double strand, and double strand then through hydrogen bond become the double helical structure. So DNA synthesis taken place in the cell like this way. So this is the building blocks of DNA, very simple form. So if you are asked to write the building blocks of DNA, this is the simple. Here you see phosphate, sugar, and then base, and then base, phosphate and sugar. In total, it is called nucleosides, and when base is plus, then it is nucleotide. And every DNA is a polymer of nucleotide. So lots of nucleotide together is called DNA polymer. GC means guanine cytosine content in every cell of organism is unique. So usually generally 50% GC out of 80 GC, but it is extremely variable among the organism. So GC content in different organism, for example, slime mold 22%, mycobacterium 73%. So it is a very unique, you can understand the information. So base pairing, already I discussed, adenine and guanine are bigger because they are both have two rings. You see adenine and guanine, two rings. Cytosine and thymine only have one ring. So this is why, this bondage, a smaller base is always paired with a bigger one. This is the, in chemistry, the affinity smaller one always like to make friendship with the bigger one. Even in our society, it is happening. Anyway, why adenine pairs with thymine and guanine pairs with cytosine? This is an interesting question. And if you are asked in your exam, you should answer these particular pairs fit exactly to form very effective hydrogen bonds with each other. These hydrogen bonds holds the two chains together and to keep the two chains at a fixed distance from each other all the way in the all along. Here you can see two. Here three hydrogen bond. So that's electron, unpaired electron, which bond here. So it is very important, and this is why adenine bind to thymine, guanine bind to cytosine, and these are the reasons. And what forces affecting the stability of DNA double helical structure? DNA double helical structure is stable structure, not it is unstable. And now question is, what are the forces help to maintain this double helical structure? Hydrophobic interactions stabilize the structure. Hydrophobic interaction. Hydrophobic interaction means dislike water. Hydro means water. So hydrophobic inside is hydrophobic. So two side is hydrophobic, but outside is hydrophilic. So hydrophobic, hydrophilic. One is repelling. Hydrophilic means attracting to the water, and this is why it is stabilized. Stacking interaction also stabilized, relatively weak, but additive van der Waals forces help for stacking the molecule. Hydrogen bonding present in the bases, they are also helping the stabilizing the structure double helical. Relatively weak, but additive and facilitate stacking. Electrostatic interactions destabilize, contributed primarily by the phosphates, affect intra-strand and inter-strand interactions. Repulsion can be neutralized with positive charges. So positive charged sodium ion or protein help here to destabilize. When DNA, open the double helical structure, electrostatic interactions is increased. So there are lots of interactions and forces exist, and overall DNA double helical structure in the cell is stable. So these are the answers what forces affecting the stability of the DNA double helical structure. At your stage, these are the simplest answer, but it is a little bit more complex. So overview of DNA structure. DNA is a double stranded helix. The two strands connected by hydrogen bond, base pairing between A to T and G to C. The DNA double helix is antiparallel, which means that the 5' end of one strand is paired with 3' end. Prime means in the sugar, there are several points in the sugar, ribose sugar. So 1, 2, 3. So these are denoted as prime, so of its complementary strand. So DNA strand, single strand is like this, and this is the templated polymerization for making the new DNA, and double strand is gradually progress like this way. So this is the summary of the structure of DNA, and all cells replicate their hereditary information by templated polymerization. So DNA polymerization, as well as synthesis of DNA, copying mechanism of DNA, which already been told by the Watson and Crick in their paper, is fundamental. Otherwise, one cell could not get the same DNA from the other cell. So how DNA translate into the protein? Because I told you, whatever we see in the living organism, they are the protein. The process is called, transcription and translation. Transcription means DNA is copied into mRNA with the aid of RNA polymerase. So when anything is copying, then polymerase enzyme, that means polymerase means to make polymer. So RNA polymerase enzyme is aided, helpful for making, copy of mRNA from the DNA. And this is called, transcription. And the RNA polymerase will bind to promoters that act as signal in the DNA sequence to make RNA. Promoter means at the point of, starting point of making the RNA in the DNA, that it is the signal. So transcription is a process of making the RNA from the DNA, copying the RNA from the DNA. So by using RNA polymerase, that means transcription process involved RNA polymerase, and transcription process result mRNA. And transcription, how transcription taken place, you can see this video. You see DNA double helical structure unzip, and then RNA is formed. Here you see when unzip, then complementary nucleotides are added, and then single stranded, this is the RNA is synthesized. So this is animation you can easily understand. So here you need to know these two terminologies, exons and introns. Exons are a segment of DNA in eukaryotic organism that code for specific amino acid. That means during transcription process, mRNA contains several segments, and also exons and introns. Exon is, involved in carrying the message of recruiting specific amino acids, but intron, a segment of DNA that does not code for an amino acid. That means in a DNA, all the segments are not carrying the gene. All the segments are not, involved in, carrying the message of amino acid synthesis. So this is the structure. So all cells transcribe a portion of their hereditary information into the same intermediary form called RNAs. That means RNA is an intermediary form that is synthesized from the DNA by using RNA polymerase. and it is called the central dogma. Very important slide for your understanding, and I made it the simplest form so that you can easily understand. Please look at here. DNA synthesis occurred, okay? DNA present in the cell. Then DNA can replicate and make lots of copies of DNA. So in case of DNA, when transcription taken place, this is the DNA, and what is happening? DNA, from DNA, we can see the through the transcription process the RNA, and this RNA is mRNA, messenger RNA. How it is formed? By the application of RNA polymerase. RNA polymerase, when DNA unzipped and then recruit the complementary nucleotide, as I showed in the previous slide, then it produces the RNA. And RNA then leaves the nucleus and moves to the ribosome. And in the ribosome, depending on the sequence, every 3 nucleotide carries information of recruiting a particular amino acid. So particular amino acid recruited, 1 amino acid, 2 amino acid, 3 amino acid, depending on this sequence, the sequence of amino acid recruited. You can ask me where amino acids are available. In the cytoplasm, amino acids are synthesized and they are present, 20 essential amino acids. So they are recruited and make a protein. And it is called translation. That means the information carrying by messenger RNA, and then in the ribosome, depending on the information, the protein is synthesized. So transcription and translation, these 2 major steps are involved in protein synthesis of the cell. And protein synthesis obviously is coming from the command from the DNA. That means DNA, from the DNA, mRNA, RNA to the protein synthesis. So this is called central dogma, and this is very important. And protein actually are the main biomolecule which regulate the traits of and functions of all the cells. Some proteins are enzymes. They are involved in all metabolic pathway, and some proteins are structural. For example, your hair, your bone, your nail, or even the cell membrane, they are the protein. So I would like to stop my lecture here, because otherwise you will be confused. So what I want to do, I want to discuss with you. Later I shall elaborate these fundamental topics in next class. So please ask me a question. Please ask a question.
Sir, what is post-transcriptional?
What is?
Post-transcriptional.
Post-transcriptional modification. Post-transcription means, after the synthesis of protein, then these protein-protein interactions and involvement of the protein to the metabolic pathway and other changes taken place. They are the post-transcriptional. That means after the transcription, after the synthesis of the protein, then the changes and reactions taken place, they are called post-transcriptional. Even protein can be modified and to use in specific purposes. Thank you very much.
Thank you, sir.
Any question? To understand biology, to understand biotechnology, you must understand the lectures that I gave you, because they are the basic pillars or basic process or basic molecules for the life, even our life. Any question? If no question, then I shall move a little bit because I have time. I shall move a little bit more because you may understood well because you attended biochemistry course. So you have another 1 minute time to ask the question, and then I shall move to the lecture. Second phase.
Sir, I have some comments.
Yeah, Tasnima, please ask question.
Sir, some comments.
Yes.
Sir, the volume of the classes looks huge, so we face some problem while reading. Sir, can you please go through a little bit slower?
You should be smart enough to understand the lecture. If you understand the lecture, you can understand the your learning points. Volume means I am offering you enough resources so that no need to go to the library because you have no way to go to the library. Hello, who is making the sound? Chinmoy, I found you are making the sound. Chinmoy Dash. I saw you are on mute. Yes. Chinmoy Dash. This is terrible, and I cannot accept this type of noise during the class because other students and I also be disturbed. Please ask some question. Afsana Jarin, please make you mute if you do not ask the question. Any question? Then I shall move a little bit on the lecture.
Sir, aske thak sir.
I have still time and I shall discuss few slides only. Thank you.
So, time
so in this, central dogma, 100% students should get the clear idea because you receive the central dogma during your biochemistry course. I am just reviewing the central dogma. Do anyone have any question on this slide? DNA to RNA is called transcription, and RNA to protein synthesis is called translation. Do anyone have any question or you do not understand anything else? So I shall show you a little more information regarding how protein is synthesized. In the RNA, several genes are present during the transcription process. The RNA is synthesized. For example, gene A, gene B, and gene C. So when they express, then specific protein is synthesized. So it carries by the RNA. And gene, is the sequence of DNA which can, express and transcribe into the RNA and responsible for certain trait of the organism, the boishishta of the organism. For example, BT gene. BT gene means the gene in the BT brinjal which can synthesize BT toxin in the plant and stop eating by the lepidopteran insect, brinjal shoot and fruit borer. That means that gene can be transferable and making the transgenic, and gene can express. But you must remember that gene expression is depend on the environment. You may have a very good gene. Everyone have, many good genes, but depending on the environment you are living, some of the genes are not expressed, so you cannot show your real potential. So expression of the gene also associated with the environment. So environment is very critical. So transcription and translation, I am giving you just like class note for your exam. DNA to mRNA transcription, and this is the simple definition. And transcription, already I showed you how it takes place. And the fragment of genetic information corresponding to one protein is one gene. So if you are asked what is a gene, a gene is a fragment of genetic information corresponding to one protein in one gene. If I ask you where gene locates in the DNA. And you see, there are genetic code in the DNA. There are three letters in every sequence of DNA carries information of recruiting one amino acids. So I shall next lecture I shall discuss on this. Three letters means three ATGC carries an information in the mRNA, which give the command in the ribosome to recruit an amino acid. So 20 amino acid, are essential for the structure of protein. So there are some rules. So the sequence here, you can see these sequences, three letters. For example, cytosine, uracil, recruit leucine, like that. So this is very, very important genetic code which carries information for recruiting particular amino acid, and one by one those particular amino acids form the protein. And every gene has the ability to synthesize one protein to do particular job. For example, BT brinjal synthesis of BT toxin. So protein and enzyme, they are protein and enzyme are same molecule. All enzymes are protein, but all proteins are not enzyme, because some proteins are structural component of the cell, but some proteins are catalytic, catalyst of biochemical pathway. You studied lots of biochemical pathway like TCA cycle or glycolysis cycle. Every step, each enzyme is involved, and enzyme is synthesized through the central dogma. So this is all about today's lecture. You have some more time to ask the question. So now the differences between DNA and RNA, their structural features, their functional features, how protein is synthesized in the cell, and why, what is gene, and involvement of gene for synthesis of a particular protein, and location, presence of the gene in the DNA. And one important point is DNA strand contains lots of nucleotide, but all the sequences are not the gene. A non-genic sequence also present there. So only genes are the functional sequence that can follow the central dogma for synthesis of a particular protein. And protein is the real biomolecule do all the job in our all the living organisms, their features, their all activities. So in a given time, you can understand, for example, in your cell now, you are listening my lecture, lots of for your attention, or you are trying to memorize and learn new things, lots of new proteins are synthesized due to memory related or learning related genes are expressed through the transcription and translation process, but they are not expressed in other type. So depending on the situation, environment of the organism, a particular gene is expressed, a particular RNA sequence is generated by the RNA polymerase, then RNA moves to the ribosome and synthesize the protein. When you are in a cold environment, then cold tolerant genes are expressed, and then you can tackle the cold or like that. When you are in the exam, new genes are synthesized to delivery or activate your memory so that you can put answer. Yes, I can see the hand from Chinmoy Das.
স্যার সরি স্যার, আমি স্যার এখানে হুট করে বৃষ্টি নেমে যাওয়াতে স্যার আমি তো বাইরে বসে ক্লাস করি স্যার, আমি একটু ঘরের ভিতরে গেছিলাম, তো ওই সময় স্যার পিচ্চি বাচ্চারা এখানে স্যার গোসল করতেছিল, মানে লাফালাফি করতেছিল, ওদের চিৎকার শোনা গেছে মনে হয় স্যার, সরি স্যার।
Thank you so much. I understood you. Yes, thank you so much. Yes, we should be, civic sense is very important. We should be careful because we are all in a remote place. So it is very hard if we are not collaborative to each other. I appreciate you.
Thank you, sir.
For your understanding. Thank you. Anyone have any question? I am, expecting some question because this biotechnology, if you do not have enough question and you cannot learn more, you can ask question related to COVID-19 also. I shall give maybe if I find time, the biotechnology of COVID-19, that is, do you know the name of the pathogen of COVID-19 disease? Who can tell me? In your quiz exam, it will be. What is the name of virus involved in COVID-19 disease?
SARS virus.
SARS virus is a very, vague term. There were SARS virus in the past also. What SARS stands for?
Severe acute respiratory virus.
No, severe acute respiratory syndrome. Syndrome, not symptom. Syndrome means multiple, plural. So severe acute respiratory syndrome, SARS, COV, COV, O small, COV2. The current COVID-19 virus name is SARS-CoV, O small and all other capital -2. That means SARS-CoV-2. SARS, severe acute respiratory syndrome coronavirus 2. You can listen the recorded video to remember it, but you can note down it. SARS, SARS -C, capital C, O smaller, and then V, CoV -2. That means severe acute respiratory syndrome coronavirus 2. Can anyone tell me what I told? Abdul Rahim, are you hearing me? Abdul Rahim.
I am hearing you, sir.
Oh, very good. So tell me the pathogen of COVID-19.
Severe acute respiratory syndrome, COV2.
Yes, COV means coronavirus 2.
Yes, sir.
There were two others coronavirus in the past, 2002 and 2013. One is SARS, severe acute respiratory syndrome only, and other one was MERS, Middle East respiratory syndrome virus. So this is the third, coronavirus affecting severely human. Any question? Can anyone briefly tell me the central dogma? If you do not tell me, I shall ask you a question. So please tell me briefly the steps of central dogma, how protein is synthesized in the cell. Ishrat Jahan Choiti, can you hear me?
Yes, sir.
Yes, tell me the two major steps involved in protein synthesis in the cell.
Central dogma is from in two steps. One is transcription and translation. Central dogma DNA synthesized from DNA. It's synthesized into mRNA by transcription, and the RNA synthesized into protein.
Yes, excellent. That means two process. One is transcription. That means synthesis of mRNA by the command of DNA in the nucleus by RNA polymerase. Transcription. And then translation means mRNA carries the message and moves out from the nucleus and goes to the ribosome, and based on the message it recruits amino acids by the use of tRNA, and then the amino acid polymer of amino acids is called a protein, and protein is synthesized through the process of translation. So two steps: transcription, translation. Transcription taken place in the nucleus, translation taken place in the cytosol at the ribosome. Clear?
Yes, sir.
Okay. Jerin Tasnim, can you hear me?
Yes, sir.
Can you tell me the chemical structural difference between DNA and RNA?
Chemical structure or structural difference, sir. Structural difference between is DNA is double stranded and RNA is single stranded.
Yes,
is,
but what are the differences between RNA and DNA in the point of chemical composition?
so in point of chemical composition, there is this fact that adenine, guanine, cytosine and thymine is present in DNA.
Are present in DNA.
Are present in DNA, yes. And sir, instead of
thymine?
Thymine, yes sir, there is
uracil is present in
RNA, yes.
RNA. Excellent, excellent. I ask one question. Khushida Jahan Mila, can you hear us?
Yes, sir.
How about the diameter of a DNA molecule? Diameter.
2 nanometer.
2 nanometer. Fantastic. That means you are nicely following.
Thank you, sir.
The lecture. Excellent. I shall ask one question to a male student to whom I should ask. Most of them are Nazmul Hassan. Can you hear me? Nazmul Hassan. Nazmul Hassan. Hasibul Hassan. Can you hear me? Hasibul? Can you hear us?
Yes, sir, I can hear.
Hello? Hasibul?
Yes, sir, I can hear, sir.
Excellent, excellent. Can you tell me how many, the nitrogen bases are used in synthesis of a DNA molecule. Nitrogen bases.
Sorry, sir, I cannot understand.
Ayesha Siddika, can you tell us? Ayesha Siddika Jerin? How many nitrogen bases are involved in synthesis of a DNA molecule? Or Akbar Khan?
Yes, sir.
Tell me, how many nitrogen
Yes, sir.
are involved in
Its answer is 4.
4. Yes, 4. What are the 4 nitrogen bases are involved in DNA synthesis?
So adenine, guanine, thymine, cytosine.
Yes.
In DNA.
Possible. Have you understood the point?
Yes, sir.
Okay. Hasibul, you have another chance to give the answer. DNA is present on the chromosome in nucleus. Are there any location in the cell where we can find the DNA? Outside the nucleus, are there any cell organelle or any location in the cell DNA is present?
Mitochondria.
Excellent. Mitochondria, and in case of plant, plastid.
Sir, plastid.
Yes, excellent. Plastid DNA and mitochondrial DNA, they are also heritable, but their size is small. So major DNA present in the cell is the nuclear DNA. When we do the genome sequences, you know, genome of an organism means, the total DNA present in the cell. So we genome sequence, obviously you have heard about it. The sequence of the DNA, that means A, T, G, C, this linear sequence, there is a machine. After extraction of the DNA from the cell, we can do the sequence. So your sequence, for example, Aisha Siddika Jerin sequence and Hasibul Hassan's DNA sequence must be different. And this is why they are, in many characteristics, they are unique and different. Clear? Last chance to ask a question. You know, your teacher is a living encyclopedia and you are living in your home. So why you are feeling shy to ask a question?
Sir.
I love the students who ask the question.
Sir, I want to ask a question.
Yes, Mo, right.
Yes, sir. Sir, you have mentioned one protein, which was the expression by BT gene. Sir, the name was skipped. Somehow skipped. I skipped this.
Okay, BT gene, Cry1 gene, when it is expressed in brinjal, as it is carrying the transgene, it synthesized a protein. Cry1 gene synthesized a protein that is, an endotoxin. Cry1, gene produce a toxin, BT toxin. It is called BT toxin. So that protein basically, an endotoxin. Many proteins are toxin. It is active. It shows its toxicity when the environment is alkaline. That means pH is higher than 7. And insect gut, pH is higher than 7. This is why insect gut it express, and gut membrane is disrupted and insect then die. But in case of human and other animals, where gut environment is acidic, our gut environment is highly acidic. This is why that protein remains silent, no toxicity. Is it clear?
Yes, sir.
Okay, excellent. Excellent question.
Thank you.
If no question, then I shall stop my lecture here. And I appreciate all of you for your cooperation. And I also sincerely wish all of you for safe and healthy stay. And please remember the guidelines of maintaining your life so that you can keep you free from the ongoing pandemic coronavirus, which is really a big threat for the humanity. And Allah save us. And uh, please take care. And within this period, before my next lecture, if you have any question, please ask the question through the Facebook. I have three good news for you. One is I shall upload a book which is 12,000 taka cost, for you in the Facebook soft copy, so you can get all the information that I am telling you, but a bit elaborate, not so concise that I am giving. I shall offer you as a gift of the Watson and Crick paper, DNA double helical structure, very simple paper. So please go through that paper, which was a Nobel winning discovery. And I shall obviously upload the YouTube lecture, which I will upload in the YouTube. So whenever you need, some of you may miss my lecture during when it is continuing due to some reason, you can rewind and repeat and listen the lecture as many times as you need. So when you visit any of the Facebook or YouTube, please make your comment. How did you feel? Because I want to deliver best service to my students, because our students are the future of our country. And the best you receive the training, the best you would be powerful in future. Thank you